This on? Good. Thank you for having me. Thanks to Propel and Bio-Rad. This is a great example. This meeting seems to be a great example of the support we've gotten over our, I think, 8 months of owning the Yeti. We have number 12, not number 1. But especially Mike, I've texted him at really weird hours, and he's always responded right away. And and he's hooked me up with whatever I needed, so it's been really good. So I'm going to talk a little bit about who we are and where we come from. This is our medical center up in the hills of New Hampshire. So we're part of the Norris Cotton Cancer Center at the Geisel School of Medicine at Dartmouth. It used to be called Dartmouth Medical School. Then Dr. Seuss's wife, whose last name is Geisel, gave a lot of money, and it's now renamed. So I'm just going to go through DartLab, a little bit about us, then Yeti Characterization, which is some of it will be similar to what you've seen from Karen. Some of it is a little different, so it'll be kind of interesting to see how different labs do it, maybe. And then I'm going to get into some sample 15-color data. So DartLab, we're a flow cytometry core facility, but we're also an immunoassay core facility. So we do a wide range of things. We keep the cytometers in use for everyone on campus who wants to use them, but we also do custom immunoassays for any PI who wants it, any clinician, and we also work with a lot of small biotech and even big pharma companies. So this is our mission statement, and it boils down to getting the right answer the first time with the least variability. So here's a list of our services. We do a lot of immunoassays for people, proliferation, phenotyping. You can make Tregs. We do a lot of multiplex cytokine analysis for people. ELISpot. We'll isolate PBMCs for you. We'll do subset enrichment. And we could do sorting, surface staining, intracellular staining. And then we do a lot of education and consulting. We run a flow course 2 times a year that has five different lectures, taking people from this is what a photon is to, here's how to run a 10-color experiment. We have, everyone comes to us and asks, how do we design our experiment? We have a good relationship with our users that they finally realize that you can't just go through your neighbor's fridge and try and put a panel together. So it's a learning process for everybody. And we'll also, we'll do consultation on flow analysis, and we'll also analyze for people if they decide to pay us to do that. And we've done a lot of clinical trials and collaborations with pharma. And I have a list of those that I'll get into in a second. We also do a lot of QC and harmonization. We participate in a lot of proficiency testing, both for flow and other immunoassays like ELISpots, multiplex cytokine analysis. We have been approved by certain agencies for doing PBMC isolation, and we're also a member of the ABRF, which is kind of the core facility national group, and we're part of the flow research group. And what we look at is some flow cytometry, effects on cell sorting, effects of cell sorting on cells for genomic analysis downstream. So here's our lineup of cytometers. We have the FACSARIAS-2 cell sorter. It used to be a FACSARIA-1, and then BD came and took its guts out and made it a 2. We have a 10-color Gallios. We have a 26-color Yeti. It kind of depends on how you count the colors, but there's 28 PMTs, I think. And we have 4 Miltenyi Max Quants, which are really kind of our workhorses. They're all 8-color. One of them has a yellow laser. People love them. They have a plate reader. No one wants to use the Gallios because they have to use tubes. So having a plate reader in the Yeti is going to move people past 8 colors, which is really nice. And then we have some, this is more of our immunoassay type stuff. We have an ELISpot reader. We have a sector imager for multiplex cytokines. We have a seahorse for cellular metabolism analysis. And it's actually Bio-Rad. I don't know why I wrote Millipore. That's where we buy the kits. So sorry, Bio-Rad. It's a Bioplex 200. Maybe we should buy your kits too. Anyways, so this is just a list of clinical trials we've been involved with. Now, not all of these are the diagnostic or endpoints of the trial. Some of them are just mechanistic studies that have gone alongside. But we've been involved with a lot of clinical trials. We've had some in a bunch in multiple sclerosis, and we've looked at cytokine analysis. We've done some multicolor phenotyping. We've looked at B regulatory cells, T regulatory cells in a few of these. And we've even done proliferation assays and ELISpot. And we're ramping up right now to do a big study with Sanofi for anti-CD40 ligand. It's kind of confidential, so don't buy or sell any stock based on this. And we're doing a big one right now. So anything basically past this line here is going to be analyzed exclusively on the Yeti, because it just gives us that much more flexibility. So I'm going to go into a little bit of what we did to characterize our Yeti, and a lot of this is kind of similar to what Karen showed and kind of different in some ways. And so it may seem redundant, but I think it's maybe interesting to see how different labs have looked at this. So just to start, we kind of don't like the way that the PMTs are named. And so there's an article from last year in Cytometry by Mario Roederer kind of describing how PMTs should maybe be named across all cytometers everywhere, and to eliminate some of the ambiguity, because FL1 on certain cytometers is, it's FITSI for people who've grown up with calibers, but it may not be FITSI. It's definitely not FITSI on the Yeti. And so that, people labeling axes with FL1, it's kind of meaningless. So what he recommends is that you have a code that includes the laser line and the bandwidth of the filter in front of the PMTs. And so what we've done is we've adapted it a little bit. He has a range for the filter, and that's just a little too complicated for our users. So we just do the laser line, and then for bandpass filters, we take the middle value, because that's kind of where people expect it to be. And then for long pass, we just add the suffix LP for where the long pass begins. And so, for the FITSI PMT, which is FL3, I think on the Yeti, our Yeti, we call it B525. So it's blue laser, and the middle of the bandpass filter is 525. And so we made this little chart for our users, and it's not really to scale. It's kind of made in Excel, but it's just a little handout so they can kind of know where where the different fluorophores lie, and it kind of gives you a vague idea of where you might have some cross laser issues. So this is our configuration. I think everyone maybe has a slightly different Yeti. We have a 5 laser version with 5 detectors off the ultraviolet, 7 off the violet, 4 off the blue, I'm counting as I speak, 7 off the yellow, and 4 off the red. And not all of these have super awesome fluorophores yet that you can buy, but we figured kind of if we got everything, we could be kind of future-proofed. And so when I said, we have a 26 color machine and we have 28 PMTs, some of that is because, this PE detector on the blue, spans both PE and PE Dazzle and PE CF594, and so you can't really use it at the same time as you're using PE and PE Dazzle on the yellow, because they'll both show up there. unless maybe you're doing things that are always on different cells. So yeah, we have that detector. And so I think this is kind of useful. We put in like our most favored fluorophores for people to use, so they can, we could just print this out and hand this to them when they need to. And we've also, I don't have a picture of it, but we've also added our Yeti into fluorofinder.com, which is a really useful website that allows you can pick your markers and tell them whether they're high abundance or low abundance on a cell, and it will kind of tell you where you may want to put them on the Yeti. And it's a pretty useful website that if you haven't seen it, you may want to check out. So we did characterization. We mostly used beads to do this, and we used a range of them. And we have some size calibration beads, which you've seen in both talks already, and you're going to see a very similar slide from me that you've seen twice already. We have 8-peak fluorescent beads, and we've also used these kind of new beads called Quantum Simply Cellular, and I'll get a little bit more into depth of what they are. So here we have the Micron calibration kit from Bangs Labs, and you can see they have 1, 3, and 6 microns. They're showing it against FITC. I think it's more interesting to show it against scatter, especially with both scatters. And we can easily see all 3 of these beads with both the regular FSC and the small particle detector. And this isn't that hard, and pretty much any cytometer, recent cytometer could probably do this. And then we went to the submicron, and again we saw this. We were able to see all 3 populations very easily. Down to 0.2. What we don't have is the 0.1 filter on our sheath. I don't think we do. I've never actually looked at that filter. But if it's a standard default, it's the 0.22 that came with the Yeti. And so, we get a little better spread here with the small particle detector, which makes sense because these are small particles. And we tried, as I guess everyone else has tried, the nanoparticles. I don't know if they've tried them from Bangs Labs or other places, but I wasn't able to see them. But now that I realized you could maybe trigger or threshold out the FITC negative, and if I get the filter on the sheath, I might be able to have a better chance of doing it. But we'll see. I'll try that when I get back. But anyways, we were happy to see that the Yeti was as capable as advertised for these beads. So we did a similar 8-peak voltage test that you heard about earlier. And we had 8-peak beads, put them in 1.5 mil of PBS just because we have to test a lot of voltages and you're running a certain amount of volume. And these are Sphero rainbow calibration particles that have this about 3 micron. You can get them from BD. And they don't expire. They last forever. So, they were pretty good. And so what we did was we acquired 15,000 beads on the stat tube with all detectors enabled from every voltage, starting at 200 millivolts, going up by 50, up to 1,000. And we had the laser wattage set to maximum. I don't know if anyone else here has even played with the laser wattage. I've left it at maximum for everything we've done. If anyone has any experiences changing it for any particular reason, I'd love to hear it maybe at the end of the talk. And then we gated singlet beads via forward and side scatter and plotted a histogram for each detector. I'm just going to show the example of the UV laser. I have a slide for every PMT, buried at the end of the talk if people want to see a particular one. So here's the UV, our first detector off the UV laser, our UV387, which is FL25, where you detect brilliant ultraviolet 395. And what I did for each of these, pardon me, was find the max number, which voltages we could see the maximum number of peaks, and what range that was in. We've seen across this range where we see the maximum number of peaks, we see pretty good CVs. It's not the exact same analysis that Karen did. That's a lot more, you have to generate a lot more numbers to do that. This is more of a qualitative analysis. But it gives you kind of a similar idea of where you're getting the best resolution of these beads, which is not always exactly where you'd get the best resolution with the fluorophore. But yeah, so for this detector, we could discern 5 peaks. But for the next one, we could get to 8 peaks, although the 8th peak is kind of smashed up against the 7th peak, but I could still, I said I could see it. And then for the next one, the BV490, where you detect BV496, saw that again, 8 peaks from 300 to 450. The next one we could get out to 6 peaks. The next one, our ultraviolet laser, we could get to 6 peaks. But again, you could see that where you get the best resolution on these beads, the voltage can be very different. This starts at 350, but when you get to this long pass off the UV, you have to get up to 600. And here's our results, which ones we could see 8 peaks and which ones we couldn't. There's an issue with this 525 in the beads. And this is something when we took delivery of the Yeti that was known to propel, and I don't remember the reason they gave me. But this is where you'd see BV510, and we've had great results with that fluorophore. So I'm not too concerned that you can't see the 8-peak beads there, but it's just something to be aware of. So yeah, I mean, you could say that these are kind of the voltage range you want to be in. If I have this data, I can go back and do the kind of sweet spot analysis to make sure that it's correct. But I think kind of for first pass qualitative assessment, it gives you a pretty good idea of where your voltages should be. So here's just that BV510 channel. And again, these are beads. They aren't stained with BV510. And what you see is everything is shifted way out, even at 200 millivolts. But I mean, you can start to get peaks, but they're all very far out. And again, I forget the reason, and maybe someone can tell me at the end of my talk what the reason was. Mike, was it Mike or Eric who told me what the reason was? One of them. So, but that's just that. So, from the 8-peak voltage test, we've done this on all of our cytometers, and we get way more peaks on the Yeti for any of these than even the Gallios, which is our previous kind of gold standard. We could get 8 peaks in the PSI7 detector, where we never, which looks like really bad on our other cytometers. I mean, we only get, I think the Gallios is decent. We get maybe 6 peaks, but the max quants, it just looks like kind of garbage. But again, antibodies look great, so it's not a true substitute. And I think this procedure can be helpful for determining a voltage starting point. It's not the be-all, end-all for that. But if you look at a dim peak and you can see where you see it, you could say, well, I don't want to go below that, because then I have no chance of seeing my antibody. But we want to do a little more in depth for some panels that we were developing for a clinical trial. And so we got with these Quantum Simply Cellular beads. And these are sold by Bang's laboratory, the same people who gave us the particle size calibration kit. And what they do is they're kind of like MESF beads, but they will work with any antibody. So they have a defined antibody binding capacity, and you have a series of 5 beads. One's blank, and then you have one that's low, then medium low, medium high, and high. And so you can use it with any fluorophore. You don't have to have a dye that can be permanently attached to a bead. So it's actually with your own antibodies, so it's a little more accurate than MESF. And so what you can do with them is when you have a set voltage and you run these beads, you can determine whether the cytometer is linear in that range for low to high expression levels. So it's really useful. And it also can tell you, you could stain beads, run them at the voltages of your cells, and go up and down and see if your spread increases to see, oh, am I getting the best resolution I can at this voltage with my exact antibody I'm using in my experiment. So here's an example. So this is a monocyte panel we were developing for a study we're doing in ALS. And what we do is this is the standard voltages. Now these are voltages that I kind of did by eye, staining cells, looking at where I thought the negative should be, where I thought the positive should be. And I wanted to see whether the cytometer was linear in that range. And so what you have with these beads is, like I said, you have 5 beads. You have a blank bead, a low binder, a medium low binder, a medium high binder, and a high binder. And again, I just want to draw your attention to this BV510, which looked awful with the rainbow beads. It looks great with these beads. I mean, we have a little bit at this voltage. The top 2 peaks are pretty close together, but obviously not everything is way off to the right. So there's something different, something special about those rainbow beads with that detector and that optical path that is screwing things up. So what you can do is you can take the MFI of each of these gates, and some of them are a little closer than others. And then you can put them into a spreadsheet that Bangs provides to determine whether you're in the linear range for that detector. And this is an experiment I was trying to see how optimal our voltages are. So I subtracted 50 millivolts, and you could see a lot of this stuff starts piling up on the axis here. But for some of them, it's okay. And so I use this to kind of adjust my voltages to try to, things that had better resolution at a lower voltage or a higher voltage with these beads, because some of this stuff is not on enough cells to really make that determination. This CX3CR1 is not necessarily that common, and so it's hard to have a low expressor, a medium expressor, and a high expressor on cells, but with these beads, it's built in. And then we ran them at an additional 50 millivolts, and for most of my voltages, this was not necessary to go up this high. Everything kind of started shifting the negatives were shifting way out, and this was a good idea that I probably had my voltages high enough. And so this is just a little example of the spreadsheet that they give you, and you can use this to get a antibody binding capacity number. So you can compare 2 samples, and you have a standard curve, and you could say, sample 1 has this many CD3 molecules or whatever, and sample 2 has this many, because you can just plot it right onto the standard curve based on the MFI. And again, the Yeti, for everything we looked at, it was at least 0.99 for linearity at all the voltages of interest. And so even when it was stuff that was too high, probably, and away from the axis where you wouldn't want your negatives that high, it was still in the linear range. So these are a nice way to test whether your detectors are linear with actual fluorophores instead of with beads that are kind of trying to mimic actual fluorophores. So yeah, we saw a linear response. We haven't tested every PMT with this because we don't have antibodies for every PMT. But for all the ones we have, it's been linear for like kind of standard voltages. And again, we haven't really used this antibody binding capacity number. It's not something that we typically are interested in, but they are available, and they can give you that number. So a little bit about some 15 color staining. And this could easily be 16-color staining, but we did it on whole blood, so we excluded a viability marker, because whole blood is, usually alive if the person's alive. So as I'm sure you're all aware, immune checkpoint molecules are, increasingly understood to play vital roles in cancer, autoimmunity, infection. It's the revolution in cancer treatment is, modulating these markers to ramp up the immune system. And then in autoimmunity, you want to go the other way. So we're developing a 15-color panel on the Yeti to analyze some of these markers. And what we did, and if any of you are not aware of it, we probably all are, but I'll just go over it. There's these papers in the cytometry journal called OMIPs, and they're basically panels that other people have developed, and they've gone through and kind of combinatorially tried a bunch of different markers to get an optimized panel. And they're optimized panels for specific instruments. And before we got the Yeti, they were of kind of limited use to us because we didn't have a lot of the detectors. But now that we have it, we have pretty much every possible detector you could have on a fluorescent machine. And so we've used them as starting points, of what antibodies to buy, because generally people have really good. There's a strict rules of how to publish these, where you have to have a lot of supplementary data showing your titrations, showing your single staining. And it's really, you can have a lot of confidence that the people who've published these have done things the right way. And so we were basing this particular panel on OMIP 36, which is an immune checkpoint expression analysis in human T cell subsets. We made a couple changes just based on, we have an in-house company that has a specialized immune checkpoint that was not looked at in here, and we wanted to include it, and they have an antibody, it's conjugated to a limited number of fluorophores, so we can't really we don't have the flexibility of moving that around and just adding it to this. So we had to make a couple changes. But it's largely based on this. And so here's the antibody list. We have these 3 BUVs for 3, 4, and 8. We use 5 violets, brilliant blue, which is really great on the Yeti. It's super bright if you haven't used it. You may not need it. FTSE might be good enough, but if you ever need a really bright marker, it's 10 times as bright as FTSE at least. And then APC, and I don't think the paper used APC Fire 750, but we found it to be very good from BioLegend. In comparison to APC Si7, it seems brighter. So we've kind of shifted everything to that every antibody we've been buying. So here's our gating strategy. This is just to get kind of naive and memory T cells. So we're just gating out some residual RBCs here. Then we're looking at singlets. And then we have a 45 stain, which we always do for whole blood. It lets you see lymphs, monos, PMNs, and kind of bigger things too. And then also these are probably red blood cells that didn't completely lyse. Here is CD45. Yeah, we use it in every whole blood panel that we do just to clean things up. I was intrigued to see about the small particle detector, how you can really, any contaminating red blood cells, you can gate them out with that. But I think we found this to be pretty useful in any whole blood analysis. Then we look at our CD3s and our CD8s and CD4s. And this compensation is just auto-comp on the Yeti. I finally figured out how to do it, and it's been really pretty slick. You hit that pause One thing I've noticed, though, if you set a run list to auto shutdown after you're done, and you get to a pause, the Yeti will shut down. So that kind of, yeah. And then I was like, oh, and then, yeah, and then you have to wait for it to shut down and start it up and then go back to your run list and start from there. So a pause shouldn't be seen, yeah. But anyways, that's kind of besides the point. And then, we gated both of these for 45 RA negative and positive. But what I realized is that with 15 colors, if you're going to do this kind of gating, it's basically, you need to make 2 to the 15th plots to look at every combination. And you don't really need to look at every combination, but if you wanted to, you'd have to look at that many plots, and no one has that time. So I decided to do some clustering analysis with FlowJo 10. And this is kind of teaching myself clustering, and I'm a very, very much a neophyte at this, and so I'll probably say the wrong thing. But this is basically what it does is it analyzes each cell to find its nearest neighbor based on every parameter that you've analyzed. And then it clusters them together to find like with like. And it generates a 2-dimensional window that it has these computed parameters that are based on, kind of how close each cell is, how similar each cell is to its neighbor. And that's kind of my immunologist understanding of it. But it's pretty slick the way it works. And FlowJo will generate a movie for you that shows the clustering. And I think I'm going to have to go here to turn it on. So it goes through. You don't actually see this while it's happening. It just, you hear your laptop's fans come on full blast, and you wait half an hour, and then this is done. The tricky part is you can't do it. The more cells you try to do it on, the longer it takes. Anything more than maybe 30,000 cells, and it's over half an hour. So, probably we're going to have to get in touch with our bioinformatics department and farm out some of this analysis onto some beefier hardware than my MacBook Pro. Yeah, well, yeah. I mean, what you have to do is, well, with this, it was nice because I only had like 35,000 CD3 events, and so I based this just on CD3s. But if I had done the whole CD45 positive population at 300,000, what you have to do is kind of downsample, which basically FlowJo runs an analysis and throws out, 90% of the cells. So you get down to a number that's reasonable to do in this clustering. And it's actually, so if you need a really rare population, you need a really powerful computer. But, that's how the clustering ends up looking. Oh, it's starting again. Anyways, oh, there we go. So this is a plot of the end result of this, which disappeared. And what you could do in FlowJo is you can overlay things that you've gated. So here, we have the light green, which are your CD4 positive, CD45 RA negative, and they all group together, but in different small clusters. And then, the RA negative, and then over here we have the CD8s, both. And those are maybe a little more distributed. The RA negatives and the RA positives are kind of down here. And so what you can do is you could see where things appear, and then you can buy an overlay, and then you can go and gate on this. And so what I did, just as a quick example, was gate on this, just these two clusters, just to see what was different about them on these two parameters, TIGIT and BTLA. And you could see these, which if you remember, are the mostly CD8 positive, CD45 RA positive, and these, which are the CD4 positive, CD45 RA positive. You get a very different pattern of expression of these two markers. You have TIGIT positive cells over here, but are mostly BTLA negative. You have slightly BTLA positive cells here, but they're TIGIT negative. And going through, and this is just a quick example, going through kind of manually and doing this. And if you do this in Cytobank, which does a similar analysis, you can color this whole plot based on each marker, and it will be bright red where there's a lot and blue where there's little. And it's a little more useful. I just don't have access to Cytobank at the moment. But I think FlowJo is improving this plugin architecture all the time, and they have other ones like Spade. And I just think this is going to be necessary as we move up, 15 colors and beyond, because otherwise it's just going to be really hard. And you can concatenate files. So if you have multiple patient samples or something, you can concatenate them, then run this analysis, and then FlowJo will add a keyword for each patient sample, and you can go and look at how the tSNE profile changes. And so you can just click through the concatenated file, and you'll see, maybe we're going to be looking at cancer patients. Maybe they're lacking, this population, and you can click through. Your healthy control has it, your cancer patient doesn't. Then you can click on that population and see what exactly is going on with all the different markers. So this population was just based on knowing that they're CD3, and it's revealed all this structure in the data that otherwise would have taken a lot of time to look for. So just on the 15-color analysis, the Yeti is our #1 machine for looking at a lot of markers, and any other by a large amount. The setup, what I really like about the setup of the software, especially, I kind of know what's going to overlap, but for users who come and they put in things, it's really nice, that screen that has all the spectrums, and you say, well, if you see here, if you use both of these, you're going to have a lot of compensation, so maybe you want to try something different. And it's really kind of intuitive in that way. And cluster analysis is more important as the number of markers are increased, I think. I think it's going to be the really, in some ways, the only way forward. Unless if your 15-color panel is 15 broad subsets, you could do that by manual gating. But if you're looking at different markers that are all going to be on one small subset, like this T-cell panel, I think the clustering is going to be really helpful. So I didn't make a pro and con slide, but I'll do it off the cuff. The pro is just like the real flexibility that we have with all these lasers and all these PMTs. I love the speed. The speed is really great. That that we can, if we hit 20,000 events, we're not like scrambling to turn down the speed, because you don't have to worry. The flexibility with what you can, your vessel is really nice. 40 tubes, you can do your 96 well plates. I don't have a robot, so I've never tried a 384, but presumably it's possible because you can click it. And then just having the stat tube too is really nice, because a lot of automated machines like the Gallios, you're still working with the carousel, even if you're looking at one tube. And having the stat tube, just zooming that in and looking at something quickly is nice. What else do I like for pros? Once I figured out how to do compensation, it's been really, really slick. And I find the numbers to be very similar to what I get in offline, post hoc compensation. And yeah, other than that, I've been pretty happy with the hardware. We've as opposed to many clogs, we've had one clog, and it was, we're not having 50 hours of use a week. So And it was 100%, I blame it on the user. And I've admonished them to do a little more filtering and resuspending of their samples before they get onto the machine. And they came back and it ran fine with this same sample prep, just a little more care. Cons, is maybe becoming a theme. There's, right now I think the software is a little behind the hardware. There's just certain little quirks when, the other problem I had with this, when you paused and things shuts down, that was kind of annoying. And then I went back to that run list and I had to actually edit the run list and like change the name before it would continue to run. And there's just little kind of quirks like that. But I mean, they're all, none of them have been real showstoppers. They're just kind of, bumps in the road, So it's, other than that, I don't, and the other thing that I think that a lot of the analysis software is caught up, but when we first got it, scaling in different software packages was kind of tricky. Flojo has caught up. Flojo 10 has caught up. Flojo 9, if you compensate, it seems to be able to figure out the bioexponential transformation, but if you don't, you can't adjust that like you can in Flojo 10. And a lot of our users are still holding tight to Flojo 9 just because they're kind of mostly Macs at Dartmouth. But I mean, maybe this will be what pushes them out of Flojo 9 to Flojo 10. We've also used a package called FlowLogic, and they seem to have, you have to kind of manually adjust some of the scaling in that. So, and I'm not sure, I think every cytometer scales differently. When we first got our Gallios, it was very tricky to analyze that data. It would always add extra empty decades to the left of all your data. So everything is shifted out and you'd get people coming, why is my data so high? It didn't look like that on the screen. But now, with Flojo 10 particularly, you open a file and it looks just like what it looked like on the acquisition page of the Yeti. So that's good. So that, things are improving. So I'm not worried that things won't improve. And it's nice to hear that this, I don't have standby mode yet, but Mike says he's going to give it to me Monday. So I'm looking forward to that because that has been a little bit, just sucking through our DI water all day. It's not a big deal because, it seems to fail gracefully if it runs out of sheath. But it'd be better if when you're not actively running it, you click a button, put it in pause, and then come right back. So that'll be nice. So those are my pros and cons off the top of my head, and there's probably others that I've forgotten about, but maybe in the panels they'll come up. And just want to acknowledge people. So we couldn't do what we do without our patients and healthy donors. We pay the healthy donors, so they usually get a 6-pack of beer, not literally, but enough money for a 6-pack of beer for 10 mils of blood, so good beer too. And DartLab, our director is here, Jackie Smith. She was the one who really pushed to get the Yeti, and she, as we were talking about earlier, Nigel was talking about, kind of thanking people for taking a chance on the Yeti. Jackie really pushed some of the faculty to go with the Yeti. We had people who were like, well, Fortessas have been out there, and BD, instruments have been out there, and she's like, we can get a better deal with this, and they seem hungrier to kind of change what's the standard in flow. And so, I think, really happy that she worked so hard to get us the Yeti. And we have 2 lab managers, one for immunoassays, Alan Bergeron, and one for flow cytometry, Gary Ward. And they both have many years of experience. And then we have 3 technologists. Obviously with these clinical trials, we have a lot of samples coming in. So we need a lot of hands, maybe more hands than just a typical flow core, because we have that other arm. And then we get funding from a bunch of people. The Cancer Center at Dartmouth, we have a COBRA grant that in a year this image won't appear because it's ended. Not any bad reason it ended. They just have a limited lifespan, and we've reached it. And then the Geisel School has helped us out. So I'll take any questions anyone has. If you want to see any, I have all the plots for the other PMTs. If you want to see what the 8 peaks look like, we can go through them all, or I'll just take any questions.
Karen's got the first question, and I know we also have a question on the WebEx, so maybe, Nicole, you can dig that up while Karen's asking.
I just had a question on the 8-peak beads on the UV, where you said the last 2 were together. It seems kind of unusual, because usually it's the dimmer ones. So did you try like color eventing on like one of that 7th and 8th peak to really see where those 2 really merged together? Or were you just kind of eyeballing the peaks?
What's interesting is if I only showed 200 to 750. I also have slides for, the higher. And, even if you ramp it up all the way, they never really split out. And I don't know if it's a function of the beads. You could, so.
So what I'm asking is, are those really 7 and 8, or is it 8 just kind of split? So if you look at like FITSI, where you can see all 8, and you color, you gate on the 8.
Yeah, that's a good idea. I haven't done that.
Okay.
Yeah, no, that would be a great idea to. Yeah, sorry, I misunderstood your question.
Did you get to his question yet?
It was a comment, not a question. So yeah, we're good.
Okay, never mind. Thanks for coming in, Kurt.
The bead question, which is so popular. And what was the proposed explanation? I mean, and also what size were they? They like 2 micron or 5 or 7?
3. So, yeah, this is with the BV510 PMT.
Yeah, it's a little disturbing.
Well, it was only that one, and do you remember what you told me, Mike?
I would expect it maybe off of the 64020 with the yellow laser, but I'm not really sure about that. We'll have to talk about that.
Yeah, I mean, it's the exact same tube. I didn't run a special tube for each PMT. It's the same PMTs at each voltage level, and that's kind of what it turned out. And then, like I said, when you run the QSC beads with, the actual antibody and fluor combination, you don't see it. You can see perfectly good negative and bright populations. So, I don't know the answer. I might try the what was the name? Cytocal? Was that the name of the bead? Yeah, I might try those to see, if they show the same. I mean, I don't think you probably didn't see it, or you would have mentioned it in that channel. Yeah. So I mean, the spheros did okay with the UVs, aside from those peaks that I'll dig into a little deeper.
I mean, you can get Cytocal beads from Cytec a lot cheaper than you can get them. A lot. Yeah, these beads are not perfect. I mean, if people die bias, they're going to have.
Yeah.
And it's so bright. I mean, those ABC beads are not going to be nearly as bright for any dye.
I'm sorry, I always have questions.
That's fine.
In your cluster analysis, how many clusters did you come up with?
Well, it's a lot.
It is, and you had 35,000 total events?
Yeah.
Yeah, so each cluster doesn't really have very many events in it, does it?
Right. I mean, these gates, this one is maybe
A couple hundred.
1,000
Yeah.
or something, and this one's a little more.
But statistically, as I told Karen at the break, I'm a big guy on minimum sampling error. And statistically, minimum sampling error is always the square root of n over n expressed as a percentage. And if you're only dealing with 1,000 cells in each of those clusters, let's say, then your error is plus or minus 3% within each cluster. And so what I'm really saying is we really need to have FlowJo working on a Cray computer so that we can put a couple million events in there.
Yeah, I mean.
And actually get some meaningful data from cluster analysis.
Yeah, like I think at this point, you know, my personal level of understanding is relatively limited beyond, you know, to kind of the tutorials that they run through. And, you know, I've read a little bit about it, and I think for my purposes now, it's maybe more a tool that can give you almost a hypothesis generating tool versus something that you could do.
Exactly, and that was going to be my next comment, because for me, cluster analysis is really kind of, frankly, a crappy way to do science. Okay, because it's like you're throwing all these antibodies up against a wall and seeing what falls out at the end. Okay, and I think a better way to really do science is really to refine your hypothesis and know what you're looking for before you go in. And obviously that's very difficult when you're dealing with unknowns. But refine that hypothesis after you find out that you were wrong, and don't be afraid to be wrong. So it's not you know, I'm not saying anything against your analysis. I would have done exactly the same thing you did, and I think it's really cool what you did. And frankly, I've got a user back in Omaha that I'm going to say, dude, you need to try this. Okay? But on the other hand, I'm not sure that's ultimately the best way to do science.
No, you know, and I think I agree. I think it's going to be, you know, an arrow in the quiver of how you look at flow data.
Yeah.
But I don't think it's going to be, unless we can get the numbers up, like you said, I don't think it's going to be necessarily something you can use to, you know, say, you know, drug B worked in this person and drug A worked in this person, and you know, it's. I think it might be a while before that is, you know, a reality. But you know, like I said, I'm very new to it. You know, I thought it was kind of cool that FlowJo added it, and I thought, well, let's see how it works. You know, rather than showing a bunch of histograms of each, you know, on each subpopulation, I thought, oh, maybe let's do something a little more exciting.
So I would disagree that it's bad science, right? So the problem that we've run into now is that this is better for discovery. And so most of the tools that we have are much better for discovery right now if you use them to their max. And so if you're using all that the Yeti can do, then you are trying to discover something upon which you can base the hypothesis. The other thing I would argue is it's not bad science, probably bad statistics. There are better methods than if we start applying Bayesian analysis to that, I think we will learn a lot more.
Well, it's incomplete science. It's incomplete because if you only did this, if you only cluster and then publish that, that's crazy. You need to get, I agree with you, you need to get to a pre-specified hypothesis and test. That's what you need to get to.
But I would argue that the data, so what Bayesian analysis does is there are no hypotheses, right? So you start, it's clean. You just have data.
Well,
and then what Bayesian does is it takes that data and it tells you what you can learn from those data. Now, it's obviously going to depend upon the number of events that you have. And so the idea that we might need to go to a Craig computer in order to be able to do it correctly. But
But even correctly, you know, you still have to, you could have, for instance, many false negatives, false positives in this analysis. You could have variability of staining that's causing that cluster.
yes.
And so that point you is great, or it gives you something you can go, this is something I never imagined before. You get to that, and you test that rigorously with the right statistics and the right methods.
And I agree with both of those. I agree totally.
Yeah, and I mean, I kind of concur with what everyone's saying. And you know, a lot of what we do in the lab is kind of trying to find biomarkers. And this is a way to maybe find a candidate and then, and then to actually develop an assay where you're looking at a specific biomarker. I don't think you're going to want to base that assay on clustering, at least as we are capable of doing it now. I think, you know, it'd be much better to have a marker you can look at in a more traditional way, everyone who wants to have a, if you want a marker, if someone's going to respond to, ipilimumab or whatever, I can't remember all the different names. You want something you can, as simple as possible, that can move to a pathology lab where they can do it on, a 5-color experiment. I think, so I guess I agree with what everyone's saying, but I do think that maybe in the future, as computing power increases, this may be more and more useful for actual, use as a biomarker, use as, making decisions about treatments. But that's just, it's kind of, I'm very much in the outskirts of the computational immunology community. So.
Okay, good discussion. Any other questions? Nobody? Are we at lunch break?